So I would like to welcome the speakers today, Ben, Michael, Rod. We also have Andrew Chapman here from the, Adam, sorry. I hope I haven't done that before. Adam from the Health Department, if you'd like to join us down here on the panel, and also Dr. Curry from the TGA, who will be joining us too, and Dr. John Layden, I've put you there too. And have I missed anyone? Have I got enough seats? Yes, I do. So I'm just going to find, I did have an extra microphone, Patrick. Oh, great. Oh, so I might just kick off proceedings, and I really welcome, this is your time to ask anything that's been burning in your brain, to if anything's come up over the presentations today that you really want to ask. Might just kick off with one question, which we just help proceedings, because we have been given some questions previously. So I guess there's quite a lot of them, and I guess maybe this one might have been answered by Ben, but there was one question here. Resection of liver metastases is proven to increase longevity. At what stage of metastatic progress is it considered timely and appropriate to undergo such surgical intervention? And is proactive early resection preferable to waiting until later stage where operation might not be possible?
I did read that one, so I had some preparation.
Oh, good. I did prepare them earlier.
So look, with liver metastases, I guess when we first see people, that's the best time to decide whether or not they have disease that can be removed. The problem is that often people have been through multiple doctors before that point and got very different stories along the way. But really the first time that I see someone is the time that I try to make a decision about whether the liver metastases can be operated on. There are some patients where we're worried about how quickly things are going to progress, and there are little things that we see from their investigations where we're worried that our treatments may not work that well. In those patients, we may wait a little longer before committing to something as big as a liver operation. And the third thing is just that I think the whole picture's changing, and we've been doing operations and then relying on other treatments to mop everything else up. But I just wonder in the future if we may move more towards an approach of peptide receptor therapy or other therapies before operating on people that may help us to pick the right patient group. So I still don't think we have the exact answer at the moment, and everything's very individualised. You've already heard of individualised story just before. I think if you've got confidence in the team that are looking after you, a decision about the timing of surgery probably doesn't matter if what's more important is when they think it should be available to you.
Thank you, Ben. Does anyone, I'm going to throw it out to the floor.
Can I just chip in one other
Oh,
comment
sorry,
from an oncology, biology side? I think that the challenge for Ben when he sees someone for the first time is that he only has one snapshot in time. And trying to make some estimate of the biology of that tumour, whether it's a good idea to take it out or whether that's not going to help in the long run. It's quite hard when you just have one scan or one biopsy, and often using time as a diagnostic tool to find out about the biology is what we're forced to do. But what we might be able to do in the future is look at the genes inside the tumour and make that decision. And so in the samples that we've analysed, there's a cluster of tumours that have a particular gene profile that just never seems to, need to relapse, doesn't grow much. And those are probably the ones that are going to behave a bit better. And so either they never need to come out at all, or they're a good candidate to get them out because they're not going to come back in the future. So I think that we're going to learn more from the biology. And then probably the other thing we can do at the moment is what Rod and the team do, which is characterise the tumour very accurately based on, that tells us something about how quickly it's going to grow based on the imaging characteristics.
One of the things that Kristen raised is the waiting and how cruel that seems for a therapy that works. Unfortunately, our hands were tied in the early days because this was considered an experimental therapy, and the only way that we could use it was under pretty strict criteria that all other treatments had failed, and that patients had to be progressing to demonstrate that they were going to, particularly if they were reasonably well, at the time they were progressing enough that the disease would likely kill them if it continued to progress at that rate, or that they had symptoms that were uncontrolled by any other way, any other means. And so under those criteria, we seemed awfully cruel to many patients, that we made them wait, earn their therapy. I think it was on a few blogs that I saw. I stopped looking at blogs a long time ago, but it did seem awfully cruel. But at this time, that's the only way we could deliver this therapy. And fortunately, we now have tools that we're better able to assess who is going to progress. We now can do the treatment much more quickly, and through the generosity of the federal and state governments of having a dedicated facility here. I had a new patient, and I think Tim saw another patient in the last week or so, who we saw for the very first time in outpatients a week before we gave them therapy, because we knew that they were going to be running into trouble if they progressed any further than they were. And so the situation has really turned around quite dramatically because of the resources that we now have and the knowledge we have. And very few patients, I hope, will have to earn their therapy in the future.
Thank you, Rod. I guess there's a question here that I might open up to, and I have given them the questions earlier, so I am not completely shocking them, but I might open up to the Health Department. Leading on from what Rod has just said, it's healthcare recognises that it needs to be evidence-based and is therefore best organised to cater for patients with common diseases, in which large clinical trials are feasible and attractive to companies because of the potential profitability if they develop a diagnostic or therapeutic approach. In rare cancers like NET, neither generally apply, and so either access is denied or costs are prohibitive. How can equality of access be achieved? And I'm going to put you on the spot because that's the $1 million question.
Look, it is. That is a good question. It's a good question for any rare cancer, for any rare condition, I think, in healthcare. Where there is difficulty in actually getting the number of patients through to provide the evidence base in some instances. And in many cases where the evidence is being generated, we'll often look to larger countries overseas, to the United States, to Europe, to Japan, to see what they've been able to identify through their own studies in a larger pool of patients. In Australia, with rare conditions, it can sometimes mean that we're drawing upon quite a comparatively small pool of patients for clinical studies. In terms of how funding can be provided to support these sorts of situations, I think we're moving to a position where the state and the Commonwealth government as well, I don't think we have a rep from the Commonwealth here today, so I won't try and speak for them, but especially in cancer, that there's a need for more flexible approaches around how funding can be provided. In Victoria, we're very fortunate that we have an entity called the Victorian Cancer Agency, which has been able to support some of the work that Rod and his team have done. There's a range of other approaches that are potentially available as well through research funding, through other supports that we can provide, such as some of the philanthropic fundraising which can be provided through this sort of group, and also through the PETA MAC in general, which places Victoria in quite an enviable position, I suppose, compared to many other places. The costs for some of the additional services, the scans, etc., are often borne directly by some of our hospitals. Over time, we would all like to see that the evidence is generated so that the funding submissions can be put up through to the Commonwealth Government to enable some of these new agents, whether it's a chemotherapeutic agent, whether it's a new scan, to actually be put up and assessed and reimbursed. That can take time depending on how much evidence is available, and it was very pleasing to hear Michael's talk earlier on the RCTs that are coming through, because that provides a very strong foundation for the assessment of the evidence. Now, whether that is actually assessed initially in Australia or whether it's assessed overseas, it doesn't necessarily matter. If the evidence is actually put up to an overseas regulator, in many instances, then that same evidence can also be provided in any submission that goes up to an Australian regulator. And what happens overseas, if it's a process that the Australian government, for instance, might consider is roughly comparable through the FDA in the States, or maybe the European regulator or European funding bodies, that can provide a very favourable sort of direction for us to take as well.
And I guess leading on to that, Rod, would you see things like the NETA-1 trial and the process, and I think Michael Hoffman would be also able to answer some of this. I think their approach with the FDA and hopefully then through the TGA and our health technology assessment as well. Is that something you see might happen quite quickly here in Australia, and will that influence also the funding for PRRT?
I think there's no doubt that randomised controlled trial data is a very powerful argument for using a therapy and to getting it approved by regulatory bodies. I would argue that the NETA-1 trial just reinforced data that had been consistently repeated by centre after centre that were doing this for a decade. It really just recapitulated. And in a group of patients, I would say, in whom the information was perhaps less relevant than the more aggressive tumours, the pancreatic, the G2 and early G3 tumours, who I've shown in my talk really benefit from this therapy in both symptoms and survival and response rate. But it's a step forward, and it is the way that I think we have to go, and we're very fortunate now from the learning that we've made around neuroendocrine tumours that we've got a whole raft of these new theranostic therapies. Michael Hoffman's doing a beautiful prospective trial in prostate cancer, a much more common disease, using lutetium PSMA. And we learned from the mistake of not collecting the data in a systematic way, not having rigorous eligibility criteria, treating patients only on compassionate grounds, and treating some patients who really, when push came to shove, were too far advanced that we would never had a chance of actually changing their outcome. And so within a clinical trial, a prospective clinical trial, we collect all the data in a systematic way. We have strict eligibility criteria, and while that might seem cruel to some patients, it's very kind to the community, because we're going to get this therapy, the data for it, much, much quicker. I wish we'd been able to do that for PRRT, but it was a rare condition. When I started this 20 years ago, the only person interested in neuroendocrine tumour in this hospital was me. It was a very, very lonely place, and now I have a whole team of fantastic people who are all incredibly excited about PRRT. A thinking surgeon like Ben, a medical oncologist who likes being in the team and recommends PRRT, and a whole group of researchers, Richard Toddle sitting down the back there and who collaborates in the genomic area with Ben. We've got now an international collaboration, and it is an international network, and so it's a very different landscape than it was 20 years ago. And I think we're very fortunate in Australia. We've got now a network of people who are interested in this. We have a special interest group in neuroendocrine tumours within the Clinical Oncology Society of Australia. And I think we're now recognised as leaders in the field. I'm on the scientific advisory board of the European Neuroendocrine Tumour Society. They used to think I was a nut. I used to stand up in the meetings and say, "You guys are crazy, you should be using PRRT." And they say, "Shut that Australian guy up." We've heard him before. Now they're inviting me to give talks at the meeting, which is a real advantage. And this year we're going to be applying, Peter Mac, to be the first European Neuroendocrine Tumour Society Centre of Excellence outside Europe. And we've been invited to apply because of the work we're doing here, which is really a great tick of health for the work that everyone here has been doing, and we're very proud of that.
Has anyone got any questions? And I'm happy to keep reading the ones that we've been given. Just on another thing, and I guess we want to recognise Katie, there you go, Linda.
I guess my question is, we're talking about the 17-year-old boy who had the PRRT, and there's obviously probably lots of people in the room who have had it. But once I guess you get to a point that you've maybe had it, and years later have it again, and you can't have any more, everyone gets excited about getting funding for it. But I guess what's next? Is there more steps they're doing overseas to look at what that next step is? What's the next exciting thing on top of that? Or is there more being done in Europe? Is there? Oh, sorry.
There's more being done here, and in this morning's session we had with the doctors, I run a preclinical program, and Adam alluded to it. We received some funding from the Victorian Cancer Agency to start to evaluate new therapies, and we pioneered this use of radiosensitizing chemotherapy with PRRT, but there are a whole raft of new therapies, and Brian Gibson over there is one of our patients who had the most remarkable response to this treatment, and because of that, he's got a particular genetic problem that impairs the ability of these cells to repair the damage that normally occurs in DNA. Something that Ben spoke about as being incredibly important. We can now, what is called phenocopy, we can recapitulate that problem using a drug. Instead of a drug that impairs the ability to repair DNA, we can block the gene protein that normally repairs DNA. And I hope no one minds that we do experiments on mice, we do. But I can cure mice with PRRT using this drug that impairs the ability to repair DNA. And that's a fabulous advance, and we're now looking to move that into clinical trials to enhance the efficacy. And that was only possible because we got money from the Victorian Cancer Agency to do these experiments. And we're now looking at a whole raft of different drugs. We're collecting tissue and trying to understand the genetics of the good responders, but more importantly, the bad responders. Sometimes we can put 100 gray or 200 gray of radiation in a tumour, it just goes like this to us and it just keeps on growing, and we don't know why. And by taking tissue and looking at it under genomics, understanding what drives it, will help us to select the next therapy and the next best therapy. And Michael also alluded to it, the immunotherapy. We're very, very keen to start to understand that intimate relationship between the neuroendocrine cell and the immune system. Neuroendocrine cells, I apologise again for my neuroendocrine tumour cells are women, but they're good at organising things, and one of the things that they do is that they sit in the bowel next to clusters of lymphocytes called Peyer's patches, which I think probably helps the body to recognise the difference between food coming into the gut, which is a protein, viruses, parasites, bacteria. It's an incredibly complex environment, and some of the lymphocytes have to start ignoring certain proteins, certain things, and that's perhaps how the neuroendocrine cells, tumour cells, evade the immune system. And if we can then understand how they talk to each other, they both share the somatostatin receptor, the T cells that Michael showed you, that had the blocks on them, the PD-1 and the PD-L1 sitting there on the cell. They're talking to one another, but the lymphocytes have somatostatin receptors, as do the neuroendocrine cells. So the same signal is interacting with both. And so as we start to deconvolve, understand the interrelationship between the immune system and these tumours, we think that's going to be the next big advance. And we're better placed here to do that, perhaps than many other places, because we're starting to do those clinical trials, and we're really excited to be involved with that. Josh Mailman sitting down the back there, he'll be getting a grant in the next week or two from us. He's involved with the NET Research Foundation in the United States, which is funding research studies, and we hope we've got a very compelling case for looking at the combination of PRRT with the immune system in both animal models and in human studies. We're uniquely placed to do that because sitting on top of us, 5 floors of research made possible by the Victorian government and the federal government building into a hospital a research division, enabling us to do this kind of research. We've got over 500 researchers dedicated to cancer in this building, and many people like me who are clinician researchers who run a lab that are able to take what we learn from patients back into the laboratory with very clever basic scientists, genomics experts and clinician, other clinician scientists. Always a very rich environment. And Joe Biden came here, he was blown away by the interactivity between the researchers and the clinicians. And I have people coming all the time from overseas, from major institutions. We had someone from Memorial Sloan Kettering, where Tim worked for 13 years, and he was just blown away by the environment of clinicians working with researchers with one goal, which is to improve outcomes of patients.
Can I just one very quickly, because I think it probably is a burning question for Katie, is if you're not being treated here at Peter Mac and you are actually, treated, and we know that the funding is coming from the VCA and the trials being done here, are there obviously open trials or these sorts of trials going on around the country with you as well, such as in New South Wales, Queensland?
There are trials. They're limited to the major net centres. Especially in what I described for the immunotherapy trials and the PRT trials, they're mainly limited to 2 or 3 sites in Australia at present. So it's really what drug companies select sites based upon, do they have the expertise in terms of the doctors there, but do they also have the large number of patients that justify opening the trial? Because opening up a trial at a hospital is very complex, requires a lot of funding, a lot of researchers, a lot of staff, nursing staff, etc. It's very expensive. So they want to make sure that they open the trials at a site which will guarantee a certain number of patients. And so that's why you'll see that some of these trials are limited to 2 or 3 cities across Australia. But I mean, it's a matter of, which is what I do, is when we have a trial opened up, we will advertise that trial to amongst all our referral base, amongst medical oncologists, across the whole state. We'll say we have this trial for this particular group of patients, and they will send, a lot of them do send patients for those trials. So it's a matter of advertising those trials across the network that we have.
Sorry, if I could just add quickly to that about the clinical trials. In Victoria and certainly in other jurisdictions, there are opportunities through many of the cancer councils to look and see which trials are actually open in a range of different cancer streams. They're often quite current as well. So it can be very good for patients to use that as a resource.
This is a question of my own, and speaking to what Katie's talking about, many of you, or many of our patients, have had multiple therapies, and Ben, this is sort of directed to you. When, with tissue biobanking and genomic analysis, we know this disease in its myriad forms changes depending on even where it metastasizes to. It also changes post different treatments. Is it worthwhile that patients, when they have subsequent treatments, have tissue further taken for analysis that may direct subsequent, maybe talking to what you're saying, Kate, like to direct their next step or help in that way? And do we know where can they do that in a third party or to analyse the genetics of that tissue? What are the costs, do you know? Because outside a trial, is this worthwhile doing?
Well, I'll start. Today I showed you some data that suggests that all of the tumours are different. They change again when they move to another place, and that's the dangerous stuff. And then when you treat them, they change again to overcome the resistance. So they change their genome to overcome the effect of the treatment. So the really important biopsy is the one when you've been through all of the treatment and you biopsy metastasis, because that's going to be different from how the cancer started. Now, in terms of what to do with that information, we still don't know, and I've made some points about directions that you could use the information we found in those particular tumours for making therapeutic choice. But it would have to be done at the time with those samples and at the moment in the setting of a clinical trial, because we don't really know what to make of that information. In terms of what's out there at the moment, and our project is, so we're moving in about a year, I think. By the time it gets all going, we're hoping to start a small set of clinical trials that will provide options for people in exactly that situation. We'll require a new biopsy and then go on to be allocated to one of several drugs. there's a few fish hooks at the moment. The first, who's going to do that and pay for that, particularly that biopsy? Unless it's a standard of care in New Zealand anyway, the government won't pay for it, neither will insurers. I'm not sure what the case would be in Australia. Someone will tell me in a moment. The other thing is that there has been a shift in the way we treat neuroendocrine tumours. So in the past, when often you've got everything growing at a similar kind of pace, and then one spot takes off or one cluster of spots. So it is now standard of care to biopsy those spots to find out what's going on inside the tumour. So at the moment, we plan to use that clinical indication, so a standard reason why a government or an insurer should pay for a biopsy to collect this tissue for that kind of decisions. And the final part of your question was about where can you send your tissue at the moment. There are some places you can send tissue. None of them provide a particularly good service, I don't think, in my opinion. They're all pretty superficial. And the genomic analyses are, occasionally you get lucky. About 5% of the time you'll find a druggable mutation that could make a really big difference in your care. But at the moment, the information that's provided is very superficial. Usually just, one or two or three gene mutations. Doesn't tell you if it involves both copies, if it's actually significant in that tumour. And so it's early days for that, unfortunately.
I think as doctors we're sometimes a bit lazy and we think we're being compassionate to our patients, not encouraging them to have a needle stuck into their liver or somewhere else to get tissue. And so we'll see a patient who had a primary tumour or a biopsy done 5-10 years ago, and they've grumbled along for 5 or 10 years with very slowly growing tumour, and all of a sudden it's taken off. It's not the same tumour. It can't be. And we've now decided collectively in our neuroendocrine tumour service that if the behaviour changes or the imaging appearances, I've shown you the FDG and the GATAID, when someone looks different, it's because they are different. They have different genetics. And so we're now going to ask patients to get a biopsy so we know what we're treating, even if it's to the point of the grade of the tumour. And so if I had advice for you as patients, is if your the tumour is changed its behaviour, ask for a biopsy. Say, is my tumour the same? Well, I was a grade 1 tumour 10 years ago or 5 years ago or last year. Am I still a grade 1 tumour if it's starting to grow? And demand it and ask them when they do it. They'll send it off pathology, but can they also put it in, if it's possible, into a biobank so that people like Richard Tothill and Ben can do the genomic analysis of when we get the funds to analyse it, or perhaps when we have the clue of what the key genes that are going to determine whether someone responds to everolimus or to sunitinib or to PRRT or whatever other therapy that might come along. I think that's going to be really important.
I think there was a question there from Grant.
Thanks. I think Ben alluded to liver metastases prognosis not being necessarily that great. Is there a particular reason why the liver is more common for metastasizing? And secondly, is it the prognosis not that great specific due to the liver, or is it just the fact that it's actually spread?
So don't get me wrong, I didn't use the right words if that's what I said. So just remember, in comparison to virtually every other cancer that I treat, I'd rather have liver metastases from a neuroendocrine cancer than pretty much anything else, maybe apart from colorectal cancer. So although the survival is not as good, it's still far better than most other cancers that we treat. And the liver is just a natural requisite for cancer in the abdominal cavity, so probably related to lymphatic drainage and drainage of the venous system in the gut, which all goes through the liver. That's probably why the liver is a common place to see metastatic disease. But also where a metastasis lands and grows needs to be in a fertile soil for it to grow. So if it ends up somewhere it doesn't like, it won't grow. And again, the liver is a very fertile place for them to grow. So so that's really why we see it. And there are many poor prognostic factors in neuroendocrine tumours. This one's just the strongest, and it's a pretty basic thing. It's just that it's metastasised, and so patients won't do as well. It's just usually the first site that we would see from that. So I don't want you walking out of here today thinking, oh my God, I've got liver metastases. That's a really bad thing. The reality is that most patients at some point end up with liver metastases. But the exciting thing is that we've got a lot of new treatments that do far better than surgery does. So the problem with surgery, which I probably didn't point about liver metastases, is that all of our tests at the moment are imperfect at telling us exactly what's going on in the liver. So I have examples of cases where we may have seen 4 or 5 liver metastases on our best scan, which is an MRI, but we found 56 when we did the pathology and looked at what we removed under the microscope. And there's probably even more than that we just don't see. So it may be that, in fact, the liver's full of little tiny tumours, and surgery is not necessarily the right way to make them, to manage them. And we need something that's going to, maybe surgery to manage the big ones, something else to manage the small ones. And I think that's the direction that we'll take in the future.
So we've got it, Catherine, then Malcolm.
Sorry, I'll just stand up so you can see me. All the things that you've spoken about today, how does that relate to pheochromocytomas and paragangliomas? Do you see when they metastasize, do they change their genomes? Are we looking at a whole other discussion that needs to be had?
Yes, so paraganglioma and pheo are actually the other area that we're particularly interested in. I started my journey a little bit with pheochromocytoma and paraganglioma at the University of Michigan, so it was actually my first love. And Richard back there, we did the first genomic landscape of paraganglioma published in the world, where we've found, and I started this project, which was also funded by the Victorian Cancer Agency, with a very naive view that a rare cancer would have a very limited genetic repertoire to get to that point, because common cancers have lots of different ways of becoming a common cancer. Lung cancer, colon cancer, breast cancers are common, and they've got lots of genes that can drive them. And I thought very simplistically, this is a rare cancer, therefore there must be very few genes that can cause it. We knew a couple of them already. We now know there's 23 different genes that, at least, is that right, Richard? 23 or 24? 21. There's a couple you haven't published yet, isn't it? Sorry, but it's incredibly complex, and they all behave differently. Some of them grow locally; they're benign in their behaviour. They don't metastasize. But there's some, particularly familiar ones called succinate dehydrogenase B, that has a much greater tendency to become malignant and to spread. But we still don't know why they do, and it's a rare subgroup of a rare cancer. And the only way that we're going to answer that is to do it internationally. And so Richard has set up with Karel Patchek and Rory Clifton-Bly at the Colling Institute, long-term collaborators of mine, a network of hospitals around the world, in the University of Michigan, the National Institutes of Health, a number of centres, major cancer centres in Asia and Australia, including the Royal Brisbane Hospital, Colling Institute, Royal North Shore, and ourselves. The American, Australian, Asian, Australasian Adrenal Alliance, A5, not the Audi, but the A5, and Ben is going to be, I think, is a member of that alliance as well, to capture the New Zealand patients. And we're going to study 50 patients who've got apparently benign succinate dehydrogenase B tumours, and 50 who've got clearly malignant ones because they've spread, to see if we can't find the genes that are driving them. That's a fundamental piece of knowledge to understanding the biology of these, but at the same time, we're treating these patients with PRRT because they express the somatostatin receptor. I think we've got, how many, Grace, you're doing the analysis? About 8 patients, 10 patients, and we have another collaboration with Hadassah Medical Centre in Jerusalem, who have another 8 or 10 patients. And as far as we know, we're one of the few centres in the world that are using PRRT. We're collecting our data, looking at the responses, and we're going to publish those results to look at the outcomes. We've started combining it with chemotherapy. Michael's, we've looked at the list of drugs that are effective in pheochromocytoma. There's not many of them, but some of them are radiosensitizers. So, you know, we're jumping in, and because there's nothing else that's working, we're collecting them. On Friday, a patient we'd treated for about 8 years has now reached the end of the road, really, in terms of the options we have. And we had a very difficult discussion with him about, you know, not having any options left. But I had a discussion with him about a program that we're running here, a unique program, I think, in the world called the Cascade program, that allows people to donate their body after death for us to collect samples of all the tissues. And many of these patients, we've done lots of imaging. We know what the images look like. We know the ones that have grown. We know the ones that responded. We know the ones that have stayed stable. To actually get those tissues and to be able to analyse them, to implant them into mice and grow them is an unbelievable tool. This patient's consented to having their tissue analysed in that way. Incredible contribution, not to their care, but to the care of patients in the future. It's really a wonderful program.
It's very welcome.
Oh, sorry. Well, first of all, just like to say what an incredible day it's been. I'm from New Zealand, Malcolm Liggett, but there's just so much hope being shown for the care of all of us, and a huge tribute to Rod and the team, but also to the Unicorn Foundation to see that wonderful collaboration, and we're certainly trying to emulate that in New Zealand. But one thing that's come out is the lack of randomised prospective data, and thankfully that is coming out now, and that is hugely important, as you've mentioned, for future funding, etc. And so the Control Nets trial was discussed. That's obviously a really important trial, and I'd just like to, could you just give us a snapshot of where that's at? And I mean, if it shows, is there going to be an interim analysis if it shows that PRRT is much better than chemo up front? I mean, when could we expect some results from that?
Yeah. So I'll start. Maybe Michael can finish. We've actually been rather poor recruiters to that trial, and even though we were very early on in the whole concept, we were, I think, probably the first people to use CAPTEM with PRRT in pancreatic neuroendocrine tumours. Perhaps Harvey would question that, but we were very early on in using it. As soon as Jonathan Strasberg had reported that trial, it seemed to me obvious to combine it with PRRT because temozolomide is a really excellent radiosensitizer. It's used with radiation of brain tumours. So it seemed to me a no-brainer to combine them, and we started doing that very early on. It's very hard when someone has been referred to your centre with the specific purpose of getting PRRT, because we're the only place that you can do it, to say, by the way, we're doing a clinical trial where you've only got a 50-50 chance of getting that therapy. You're going to get chemotherapy or PRRT with chemotherapy. And we find that a very difficult discussion to have with patients in our own mind. So we have our own patients we've been following that progress, and we think that we can put them on it. But it's very hard when someone comes with the expectation of receiving a particular form of therapy as our current standard of care. Perhaps Michael might comment.
No, I agree with Rod. It's actually a very hard question to ask patients if they're particularly referred for PRRT. So we're trying to recruit our internal patients. I gather it's open at the major PRRT sites across Australia. I'm not quite sure the exact, how many patients have I got? I've got about 28. Yeah, about 20 to 30 patients. They've got seed funding from the Unicorn Foundation for the first part of the trial, and in their process of now trying to get government funding through the NHMRC. And this is their third attempt to try and get a grant approved for the grant for their trial. So if it gets knocked back again, I'm not quite sure what.
It has.
Yeah, we haven't heard officially. I thought so.
Sorry, that's
Oh, whoops. We'll just cut that from the edit, shall we?
And there, I mean, I'm aware of moves in Western Australia, for example, that the only people who get PRRT are people who are, willing to enter into the clinical trial. We haven't got to that stage, and I hope we never do, because we'd always like to think that this will be available to patients when they need it.
Can I just a reprise to all of that? I mean, I think the thing that's really important for you as patients to realise, especially if you're on the Control Nets trial, I know some of you are, I met a couple before, is that we have what we call equipoise. So we don't know if, I mean, the standard of care at Peter Mac is chemotherapy with PRRT, but we don't know that that's better than just PRRT. Otherwise, we wouldn't be running the trial. So we don't know that yet, and oncology is full of things where we've thought this is the best thing, and then you do the randomised trial and you find out that there's no better than doing half of it. So I don't want to if you're in another centre, I don't want people to think that you're missing out on something, because we don't know if you are. That's why we're doing the clinical trial. It's a really important thing to.
There is crossover, isn't there, in the trial if there's progress.
Not as part of the trial. You're allowed to have other treatment after that, but and just in terms of the trial, so the trial as it was initially designed was for about 140 people, 160 people, I think, and we couldn't get funding for the whole thing. Unicorn Foundation got things going with
$300,000.
$300,000, which is amazing, and so enough to get it going in several Australian sites. We decided not to use any of that money to get it going in New Zealand because there was some extra costs in getting it set up. But to answer your question about the interim analysis, I guess pragmatically there's going to have to be something done with the data as collected if we're unable to get MHRC funding. But it wouldn't be a planned interim analysis because it wouldn't even be halfway through. Yeah.
So I think it's an important issue that when you do see your oncologist, to ask them what clinical trials they do have for NET patients, or to be considered for clinical trials at other sites. If you come to an equipoise where you need to consider changing treatment or starting a new treatment, that's really the only way we can answer questions where, as Ben said, we are at equipoise. We don't know which is the best treatment. Is everolimus better than PRRT? Is chemotherapy better than PRRT? The only way you can answer these questions properly is with clinical trials.
Hi.
And then I'm going to get to you.
I have a few questions, and they probably going to sound a bit selfish, but firstly, the first question is, am I living in the wrong state? Should I rather be living in Victoria to access this level of expertise? The second one is quite personal because we speak of liver metastases, but what about when the metastases goes further into the bones? What sort of regime is recommended for that? And what sort of prognosis could one expect in that situation?
What state do you live in?
Queensland.
So I'm very pleased to say that one of my former fellows is moving to the Royal Brisbane Hospital, and he's taking all our...
I've met him, thank you.
Interest and expertise with him. He's very keen to continue the Peter Mac tradition and protocols there. He's a very smart young guy, and I'm sure he'll have a really great career in this area. They've already been doing some very good work in the Royal Brisbane Hospital. It's one of the centres that's doing PRRT, and we are a small community, and we all do talk to one another, and the Unicorn Foundation has been fantastic in helping us to network and to recognise the other people who are working in the field. And I'd hope that wherever you are in Australia, you're getting good advice and good access. All the people in Adelaide came here to do some training with us. They understand our protocols. They regularly ring me or other members of the team to ask our advice. It is an incredibly well-connected community, and I hope that you will get good advice wherever you are in Australia. We treat all the patients from Tassie because they don't have a centre there at the moment, but they've got lots of clinicians who we've worked with for many years. Some come and do their sabbaticals here, and we regularly go down and talk to them, and so we manage the patients as a team. And so I'd hope that your interest in getting the best advice will be available wherever you are in Australia, and I think we're much better placed in that than perhaps any other country in the world, because we are a small community.
Look, if I could just interject there, I have had only positive experiences, but when the hard questions are asked, like, my tumors I have a partial response here, and some are shrinking and some are growing, so let's do some biopsies. And then they say, no. And then they say, biopsies won't really matter because, you were diagnosed with grade 1s and grade 2s, so that's what we are treating. And those are questions I have asked almost 2 years ago already,
Yeah.
And secondly, I think when it comes to the multidisciplinary support teams that are supposedly active in various hospitals and centres around Australia, what exactly does a multidisciplinary team consist of? Because I find that I have to go work outside of the system by asking my GP for referrals to an endocrinologist and a gastroenterologist and a hepatologist and Lord knows what. And then I say, please copy Dr. So-and-so and Dr. So-and-so, just to speed up this process, because as far as I'm concerned, I'm running out of time.
Yeah, sure. I mean, one of the reasons that we're going through this ENETS Centre of Excellence approach is because they demand a multidisciplinary team of endocrinologists, gastroenterologists, pathologists, surgeons, clinicians who are involved in this. And this has been generously sponsored, our application generously sponsored by Ibsen, with the view that if we meet the criteria, we will become part of the auditing team for other centres around Australia to get these processes set up, the tumour boards, biobanking, clinical trials, research to happen wherever. So patients aren't left with the one medical oncologist who said, I've never seen one of these diseases, go home and die. That shouldn't be the way that the world works these days. And I think that we know a lot more about this disease. We have excellent results in patients with bone. PRRT doesn't necessarily only work in the liver, only in lymph nodes. It works in wherever it is, but sometimes the target's lost and we need to be thinking about the next therapy. And one of the great things in working in an integrated service, that sometimes we're doing surgery of an individual lesion that's got a different appearance. Sometimes we're cutting off its blood supply. Sometimes we're sticking a needle into it and frying it. Sometimes we're giving chemotherapy. Sometimes we're giving everolimus for a period of time, and they change their features. And then we can go back to PRRT. I think one of the patients in the room today has been through 4 or 5 different therapies at different time, and each of them independently has worked, and a new cohort of cells has come out. It's like whack-a-mole, that game that kids play. You hit it there and it pops up here, and we just have to keep pumping it down. And you can only do that as a team, because none of our therapies independently are going to cure every last cell. And so, I think we have to keep working as a team and be really reactive and know as much about the enemy as we can. And that's biopsies and genomics in my book.
Thank you. Oh.
Just speaking to that point, unfortunately, as indicated by Kate, your story is not isolated. You are your own case manager oftentimes, even if you are seen at a centre a centre of expertise, such as Royal Brisbane or Queen Elizabeth or wherever. However, it is a frustrating aspect of the disease that you are your own case manager oftentimes, and that is collating your own information and commonly spewing it forth every time you go to see your outpatient oncologist or what have you. So unfortunately, your own advocacy is still part of this. We haven't been able to minimise that yet, even though our aspirations are true and worthy. We still have a long way to go in what you're talking about, and I understand your frustration, and it is, everyone could put up their hand and feel that in the room.
I'm generalizing now, but most would feel like me certainly Dr. Ben Lawrence could benefit by analysing my tumours as they are progressing right now. And I'm prepared to do that. I'm not quite ready for your cascade.
No.
We are all in this together. We're not fighting each other. We want to work with each other. We want to speed things up. For crying out loud, trials get to us when, after breast cancer trials are done, and that's done, and that's done. When are the next cancer trials going to happen?
We're trying our best.
No.
With what we've got. I know that might not. We might have to move on, but I know
We need
it's
to change
frustrating.
that paradigm. Trying our best is not always the best.
We need Steve Jobs to have donated.
That would have been good.
I'll sort him out once
So I thank you, and it is, and we thank you for your passion, because it is exactly mirrored across the board, and we do understand, and we do understand the frustrations, and it's the frustrations held by all of us. I do see that we've also run completely over time, and I do have one more question. I know that you've been so patient here, so we'll just take one more question, and I know that thank you for your patience, because we have run over time.
It's a quick one, but thank you, everybody. Use microphone. Can you use the microphone?
Is it on?
Yes.
Sorry. I just want to ask a quick question. Kate put up a slide today which is talking about your everyday experiences and things that you actually have to control, which is symptom control. And I was reading about Telefast, that it's got an extension by the FDA in the States, and I was wondering if you can give us an update on what that means, or if there's anything else happening as far as symptom control here.
Yeah, for those of you who don't know what Telefast is, there's an enzyme, and this is only relevant to the carcinoid syndrome, that converts tryptophan to serotonin, which is the end, that's the gossip chemical that makes everything happen in carcinoid syndrome. And by blocking the conversion of tryptophan to serotonin, you dramatically reduce serotonin levels, and many of the symptoms related to carcinoid are related to that excess serotonin. So it reduces flushing, and it particularly, the trial is finished, it's shown unequivocally that it reduces the number of bowel actions and the severity of bowel actions, which is a great symptomatic benefit to many patients. We have a card in Victoria called PTV, it stands for Public Transport Victoria. Many carcinoid patients know Public Toilets Victoria because they chart their way to Peter Mac by public toilet to public toilet, stopping many times on the way. And so controlling those symptoms is very important in terms of quality of life. It probably is also going to reduce the incidence of the fibrosis that Ben encounters when he operates on these small bowel neuroendocrine tumours, and may stop them ever needing to be resected, or the complications. The cardiac disease that was also mentioned, it may delay that and prevent the valves from fibrosing up and leaking, often a life-defining complication of this disease. And so they're really great things, but it does nothing to the growth of the tumour. And so it's just it's a palliative treatment that I think will be really valuable in combination with somatostatin analogues and with effective therapies to control tumour growth. It's very likely that this will get through the PBAC, I think, based on having a very large international randomised control trial showing significant advantages. It's been fast-tracked to the FDA, so it's going to likely be approved there. And Australia is now increasingly recognising drugs that have been approved in other regulatory environments, which is a great thing. We really struggle with PRRT, though, because it doesn't come under PBAC. It's not seen as a therapy. Crazily enough, it's under something called MSAC, the Medicare Services. It's seen as a device. And so people who are thinking about, do I fund this X-ray or that X-ray or this scan or this device or whatever, are making decisions about a therapy. It's the only therapy we know of that doesn't go through the Pharmaceutical Benefits Association, And the skill set, the expertise, and the way that they look at this is completely different to PBAC. Michael sat on PBAC for a number of years, I think. Still do? ADEC, yeah, Australian Drug Evaluation. Yeah, but the whole process of getting these therapies through is really rather poorly coordinated. And one of the challenges we have, you've got the TGA regulating therapeutic goods. How does this fit into that environment? How do we get it funded? How do we integrate this with all the other things that are being pushed forward? The drug companies are going to be have pushed successfully for some of these drugs. Everolimus is now funded for pancreatic neuroendocrine, as is sunitinib. There's applications to move that into bronchial and other neuroendocrine tumours. Lanreotide for tumour suppression. Companies have deep pockets and they can do this very effectively. I'm sitting here at Peter Mac and I have no one to put in an application for PRRT unless I do it myself. And that's really, really tough. So we need to get a better way, and that's something that Adam and I have been talking about for a long time. I'm passionate about policy with evidence and evidence-based medicine, but it's incredibly hard in this field to do that. And how do we organise ourselves to do it, I think, is a real challenge.
So,
yeah, you have to, for a submission to approval for funding, you have to actually have a sponsor, which is usually the pharmaceutical company, to sponsor the drug through the approval process, look in terms of efficacy, safety, and then finally funded by the PBAC on the PBS system. And it can take, one, it's quite expensive. Two, Australia is the backlog because we're often last in the list of patients where the company will make a submission. Three, the submission process, when they start submitting, and by the time it actually gets PBAC approval, if it gets that, might be up to 2.5 years. So it's actually quite a prolonged process compared to the FDA.
And I think this might have to be the last question. I'm so sorry because we have gone completely over time, and I'm conscious that everyone's probably getting very tired.
Under what circumstances do you ignore FDA approval of drugs and therapies and then want to reinvent the wheel again before the drugs are made available in the Australian market?
So the
Same with European approvals to drugs and treatments.
So look, the pharmaceutical industry is a globalised industry, and regulation is globalised and is essentially harmonised, has been harmonised for 20 years. So our standards in Australia are pretty much the same as the US and Europe.
It should be a no-brainer. You should just accept the other agencies, regulatory agencies' approval, isn't it? So then why do we reinvent the wheel and go through this expensive process again?
The problem is I think that this particular product is not registered overseas yet, but once it is, it would certainly go very quickly in Australia, or they would.
What's wrong with it? We have enough evidence that it works.
Well, I think that's the issue, is the standards of evidence. So I think it's not registered either with the FDA or the EMA.
It doesn't matter. We have a panel of experts there who are saying it works. So prove the darn thing.
We'd like to approve it.
We would like to
approve it. We need, as someone else said. Well, first of all,
it hasn't been approved in FDA yet either. It hasn't actually been approved on the FDA. Because there hasn't been a submission made. They can't approve something that might not have been put on. So look, this is a very, very important issue, and it's one that the Unicorn Foundation is very, very heavily involved in. We're very much wanting to put that, the HTA system is something that we're very, very heavily involved in. And look, we can actually catch up offline on that one. But I would like to now please thank all of our speakers who have given up their Sunday today to be here. And thank you to all of you for being here as well. I know some of you have travelled far afield from New Zealand, Queensland, Perth. So thank you all for being here, and safe travels home, and please, we'll keep you up to date. These recordings will be made available on our website very soon. no pressure, Greg, at all. And we, as I said, this is something that we want to continue. We'll be sending out a bit of a evaluation survey, but please have a lovely rest of your weekend, what's left of it, and we look forward to seeing you again soon. Thank you.